All right, biologists, let's take a look at crossing over and recombinant gametes. Now, consider two characteristics determined by two different pairs of genes located on the same pair of autosomal chromosomes. This is the same as saying that the genes are linked. It doesn't have to be autosomal chromosomes, but if two genes are on the same chromosome, we say that they are linked, as exemplified in the diagram here. So we have a pair of autosomal chromosomes. These are the same chromosomes from the same pair. They are homologous. Now, assume that 500 germ cells, so 500 germ cells, this is an important word, with the same genotype as the cell above, enter meiosis, and that 20% of these cells show crossing over. Crossing over is something that actually happens to all cells that undergo meiosis. This happens during prophase 1. So let's say we have this chromosome here and this chromosome here. What happens during prophase 1? What happens during crossing over is, first, these chromosomes will be replicated, right? Because of phase S during interphase. So you have this chromosome, and this chromosome. Looks like 4, but they're actually 2 chromosomes. They are just duplicated, right? So this is a double version of this one. And the blue here is the same as this one, but duplicated. During prophase 1, you have crossing over non-sister chromatids. That means chromatids of different chromosomes from the same pair, right? So we have here this chromatid, this side, and this other side. They're not sisters because they're not here the same color. They're not part of the same chromosome. They are from different chromosomes of the same pair. They will cross over each other and then separate like this. After they separate, this chromatid has one part of the other one, and the red chromatid has a part of the blue one. This point of contact here is called chiasma, right? So this is singular, one chiasma, two chiasmata. The question says only 20% of these cells show crossing over because the rate of recombination depends on the distance between two loci. In this example, locus A and locus D are very close. Even though the chromatid went through a crossing over event, because those two loci are so close, they did not exchange between the two chromosomes. But locus B here was involved in the crossing over. It doesn't mean that loci that are very close to each other never undergo crossing over. It just means that the chance of a chiasma being formed between very close loci is much smaller than between two loci that are far apart. So what types of gametes can this germ cell here form? Well, it can form 4 types of gametes. Let's take a look. These are the 4 possibilities of gametes that will come out of this germ cell after meiosis. Because the entirely red and entirely blue chromatids appear, these chromosomes, they appear here in the parental cell, we can call them parental chromosomes. And these 2 chromosomes that did not exist in the mother cell, they are recombinant. What about these 20% here? Well, 20% of these cells are recombinant. That means 10% each. And 80% of the cells are parental. That means 40% each. Now back to the question. How many recombinant gametes are we expecting at the end of meiosis? Well, I had 500 germ cells. They are going to form 2,000 gametes. How do I know that? Because each germ cell gives origin to 4 daughter cells, to 4 gametes. So 500 times 4, that's 200 gametes, but 20% of those are recombinant. So 20% of 2,000, well, that is 400. Letter B, 400.